Okay, so next we will have our proceedings presentation. Let me see this one. Excellent. So yes, so our proceedings presentation is titled Enumerating Consistent Subgraphs of Directed Acyclic Graphs, an Insight into Biomedical Ontologies. Yisu.
Thank you. Hi, thank you everyone. Hi, I'm Yisu, also known as Sean. Here I'm going to give a talk about our studies on the biomedical ontologies. Now let's begin with the protein function prediction task, which takes a protein sequence as an input and predicting a subset of the biological terms as an output. These terms coming from an underlying ontology with hierarchical structures, a directed acyclic graph or DAG. Here, for example, we have the cell differentiation term, which is a type of cellular development process. So we have the cellular development process as a parent in the graph for the cell differentiation term. Because of such structure, when we're making a prediction, we are actually making a consistent subgraph here, for example, we have the apoptotic process and the cell differentiation term for this example sequence, and we're also making prediction on all its ancestors. And to make such prediction, nowadays researchers tend to use machine learning techniques. Besides the reference annotation data, we may also consider many other types of data. For example, the protein three-dimensional structure, protein interaction network, or gene co-expression data. However, this is a very hard task. Till now, we still don't have a very good method to solve this. Here is the result from the KAPHA 2 competition held in 2013. The paper was published by John and many other researchers in 2016. First, let's see the results on the molecular function. Here, the best method can achieve about predict correctly on 60% terms. Well, on the biological process, we are doing much worse. We only predict about 37% terms correctly. Well, on the cellular component, it's a little better than the biological process. It's about 37% term correctly predicted. But the problem is that we're not doing better than the naive baseline, which is just predicting the frequency in the database of each notation. And so now we are wondering why this task is so hard to solve, and what makes the molecular function seems a little easier than others. If we look at these ontologies, we will find they are super complex ones. Here we only show the ontologies truncated at level 4 for the minimum depth of each vertex. On the left is the number of vertices and edges at the level 4 for each ontology. And for example, for molecular function, we have 600 vertices and 700 edges at level 4. And on the right-hand side, we're showing how many nodes going to be added at each level. At level 6 of the molecular function ontology, we are going to have more than 4,000 nodes to be added. Here we may notice some differences between these ontologies. First, that we notice that the biological process ontology is the most huge one, and which have thousands of vertices and edges. And also the molecular function ontology is more like a tree compared to the biological process and the cellular component. Also, the cellular component ontology has the vertices more evenly spread out through each levels. And now we may wondering two questions. First is, how large is the output space defined by these ontologies, which is the question that we're trying to answer in this work. And the second question is, are the size and the structure of the ontology constructed somehow correlated with the difficulty of the protein function prediction task? Now we want to compare our problem with ordinary classification tasks. The first most basic one is the binary classification, which have only 2 outputs. And for the multi-class case, we have several classes to choose from. And for multi-label case, we have 2 to the power of n number of combinations to choose from. Here in our problem, we call it structured output learning problem. And what's the answer? What's the output space size for this? Will not where other works to give the answer. And we may see such tasks appear in many other areas, such as the text mining or computer vision. Our finding in this work may also be applied to these areas. So what do we mean by structured output learning exactly? As we know, these ontologies are formulated as graphs, more specifically directed acyclic graphs, where the terms or concepts are represented by the vertices, and the relationships between these vertices, such as is a part of or regulates, are represented by the edges. Here in this work, we only consider those relationships that are transitive. And with such structure, its output is a consistent graph that in a subgraph, for each vertex is parent from the original graph also need to be in the subgraph. For example, here on top, the first subgraph is consistent, while the second is not, because the vertex 2 is missing. And now we ask a question that how many consistent subgraphs are there for this example that? And the trivial bound is 2 to the number of leaves, 2 to the number of vertices. And the trivial algorithm to find this is to check all these 2 to the number of vertices subgraphs to find the consistent ones. However, such an algorithm is not efficient enough, not even on 100 graph, a graph with 100 vertices. So we need a smart algorithm. First, let's check out the tree case. The algorithm for this is provided by Ruski in 1981. Here we made a small modification that we consider the empty subgraph is also consistent, which is different from the original Ruski's algorithm. And to get the count for the tree, we just multiply together the count for each subtree and add one. And for each subtree, we use this algorithm recursively. And finally, we will get the count for this for the whole tree. And if we have several disconnect trees, we will multiply together the count for each tree. However, on that case, it won't be so easy to solve. So our algorithm first pick every arbitrary node vertex as a pivot, and then split the problem into 2 sub-problems. The first one is the subgraph without all descendants of the vertex U and itself, as shown as the yellow part removed. And the second sub-problem is to remove all its ancestors and itself, as shown as the blue part. And here we may notice that there are some part of the graph is repeated in both graphs subproblems. So we may introduce some hash method to improve the performance later. Here is the basic algorithm formulated. And first is check whether it's a forest. Otherwise, we will pick a arbitrary pivot and sum together this number of these subproblems. Here, let me show you an intuition behind this algorithm. So first, with any given pivot U, and we will split the set of the consistent subgraphs into two subsets. The first set includes all the subgraphs that include vertex U, and thus it does not include the vertex U, and thus all its descendants cannot be included either. So we remove all the descendants to get the subproblem, and in the second one, all the subproblems, all the subgraphs include the vertex U, and thus all its ancestors also need to be included. And at last, we sum the two numbers together. And however, such an algorithm is not, the basic algorithm is not efficient enough to run on the true ontology. So we introduce some improvements. The first one is the strategy to choose the pivot, which has a great impact on the runtime. Here in today's work, we explored several heuristic strategies to improve the performance. Besides the pivot strategy, we also include several other techniques, such as pruning disconnected parts, or hashing the computed subproblems, or try the reverse graph, whether it's easier to solve. At last, with all these modules added and the maximum degree pivot strategy, we achieved less than 1 second for graph with 100 vertices, which is 3,000 times faster than the basic algorithm. And then we turn back to our true ontologies. Here we showing the upper bound and lower bound with the colored boxes. And the upper bound is calculated by converting the graphs into trees that by randomly keeping only one incoming edge for each vertex. And the lower bound is 2 to the power of leaves, as previously. And here, first on the molecular function, we get to the next level, and it's actually a huge number. It's 10 to the power of 2600. That's really big, and that's 2600 digits. And we did not finish to the last level previous to this paper be submitted. However, we can see that it's really close to the final number. And on the molecular function, on the biological process, we are doing much worse. We will only get to the 5th level. And we may also notice that the upper bound and the lower bound on the biological process is really huge. It's different in 2,000 number digits. And on cellular component ontology and human phenotype ontology, we get to the 8th level and the 6th level, respectively. We also notice that there are many terms in these ontologies that's never used in any annotation. So we remove them and show the removed part as show the remaining part as the light color. And we may notice that the count between the full ontology and the used parts are really also huge. Next, we think that this kind of result, is somewhat matches to the results from KAPHA 2 results. Where on the biological process, we get only to the 5th level, and we're also doing worse in the KAPHA 2 computation. And in cellular component, it's also a little worse than the molecular function. So we may naturally think whether this is some relationship between the complexity of these ontologies toward the difficulty of the prediction task. by the complexity of the graphs, we will not only mean the number of vertices or the number of edges, we also mean the actual structure of these ontologies. And finally, we check the entropy of the observed space and compare it to the maximum entropy of the these defined by these ontologies. The entropy of the observed space, we mean that we use this formula to calculate, and with the probability of each annotation from the database, and for the maximum entropy, we assign the equal probability for each possible annotation. And here is the comparison ratio between the entropy of the observed space to the maximum entropy. And we think that because of such ratio, we think it's really highly ordered of the observed space. Finally, to conclude, first we formalized the protein function prediction task as a structured output learning problem. And we proposed the algorithm to compute the size of the output space. And we found that the space defined by these ontologies is huge, and a considerable part of the space is never observed. And the observed part is also highly ordered. Based on these observations, we want to bring forth two discussions. One is that whether can we use such algorithm as a ontology quality assurance, and how are we going to use such ontologies with them going to become so huge? At last, I want to thank all the members from Peja's lab, and especially Yuxiang and Peja providing many key ideas to this paper. At last, I'm going to leave you with this huge number from the molecular function ontology at level 5. And thank you very much.
Yes, Robert.
Yeah, so I have one question. Thank you for this excellent talk. I think there are two somewhat different issues which affect the performance of function prediction methods. One is how hard the problem is biologically. And the second one is how hard or how well our algorithms are suited to the actual data and to the output space. What you are measuring is going to affect the algorithms that we use, because if we have such a large output space, of course we will need some very smart algorithms to somehow reduce this, and we need to be able to maybe use background knowledge or do something clever. There's a second issue, and this is the biological question. So molecular functions generally can be derived from just looking at a single protein. You look at one protein because it's whether it binds something or not. Is it can it metabolize this or not? A biological process will always require more than one protein. It's kind of the dividing line between them. And that makes it incredibly hard, because you do not only you cannot just look at one protein sequence and predict whether or not it participates in this process. You also need to know what other proteins are there and what their functions are. And this makes it biologically very hard.
Yeah.
And there's a correlation. And my question is, how do you see, in terms of function prediction methods, how much work do you think needs to be put in the algorithms, and how much needs to be put in better understanding the biology in order to further improve this? So what is this ratio? Where is the big limitation in your view now?
Thank you very much for the question. That's a very good question. So, as Robert said, that we may notice that the molecular function is is more direct biological terms, and well, the biological process is much abstracted. but here we may also notice that the cellular component is also very created to biological, very good. But we don't have good results on the KAPHA2 computation. So this is that we compare the molecular function and cellular component to the biological process. Well, in this world, we're not measuring how the to what kind of these terms is biologically related. Does that answer your question? Thanks. Sorry.
Are there any other questions? No, very good. Thank you very much for this presentation.